So today I'm going to talk about how we are using the TWIST panel to enable clinical trials and translational studies in Cancer Molecular Diagnostics Laboratory. So our laboratory is located in the Cambridge Biomedical Campus in the Clifford Albert building. We are a team of 12 people, and we are funded by all these organizations here, the main funder being the Cancer Research UK, Cambridge Cancer Centre. We are also part of the Integrated Cancer Medicine program of the University of Cambridge, which wants to revolutionize the cancer care of patients by using all these modalities here in cancer treatment. And two of the important modalities shown here, the tumor genomics and liquid biopsies, are handled by our laboratory. In this talk, I'll be going through the three panels which we are using from TWIST Biosciences, all of them capture-based for three different trials which are currently ongoing in our laboratory. The first one is the 350 genes pan-cancer panel. The other is the 56 genes myeloid panel, which we are doing with in collaboration with Department of Haematology of University of Cambridge. And the panel which we are developing at the moment is a 150 genes lymphoma panel. The 350 genes cancer panel is being used for the Basket of Baskets trial, which is a trial initiated by Cancer Core Europe, which are 7 countries in Europe which have started a consortium. And it's an early phase trial for solid cancers, which in the first part of the trial is to profile cancer patients for common tumor mutations. And based on the genetic mutations detected, they will be put in a therapeutic basket, which will target that particular mutation. The first module which is opened in this trial is the Immunogenetics Module, which is funded by Roche. And the way it works is that the tumor samples are sequenced with the Cancer Core Europe panel, which is our CMDL 350 panel, simultaneously with the foundation panel of Roche. And then based on the mutations detected, they are distributed into these different arms of the basket. The idea is to profile around 1,000 patients in 2 years, and then to put around 100 patients in these different baskets. So all the centers send their tumor as well as germline DNA to the Cambridge laboratory, to us, where we sequence them with the 350 gene capture-based panel. Once the results are analyzed, they are discussed in a virtual molecular tumor board portal, where all the clinicians from all the sites participate, and then the patients are put into different baskets. Like I said before, at the moment, only the immunogenetics module is open, but these are other modules will open in coming months. So this is just some QC data of how many samples we have tested with the TWIST 350 panel. And we started using the TWIST panel in summer of 2019, and we have tested around 837 patient samples. Not all of them are from the Cancer Core Europe trial. We also tried some circulating tumor DNA samples, and we standardized the method to work with around 70 nanograms of FFPE DNA and 30 nanograms of ctDNA. We had very high success rate with FFPE DNA, with 98% of samples successful, with our cutoff of mean average coverage of 200. And we have set the variant allele frequency cutoff at 3%. Ezequiel, who will be talking after me, will be going through more the QC parameters of this panel. But this is just to show that when there were samples with less than 70 nanograms of DNA, we could also sequence them successfully, and only 2 out of these 25 have failed. And surprisingly, these were not the ones which had less than 20 nanograms of DNA. The concentrations of samples which had less than 70 nanograms of total DNA extracted is shown here, and they hover around less than 1.5 nanograms per microliter. This table here shows the samples which failed our final QC of achieving mean target coverage of 200, and the samples in the 7 samples here above had much more than 70 nanograms of DNA isolated, but they still failed the QC. And we have found by doing our QC steps in the library preparation method, that the pre-capture ligation-mediated PCR QC is an important metric, because if this value is less than 20 nanograms per microliter, the samples invariably fail sequencing. Ezequiel will go through this in more details, but I'll now shift to our, just to tell you about the ctDNAs which we have sequenced with this panel, and all the ctDNA samples have been very successful with the 350 panel, and the concentrations normally hover around 1.5 nanograms per microliter. Majority of them were at 30 nanograms concentration, but they all passed the mean target coverage of 200, and they had also very high on target percentage. So now shifting to the lymphoma panel, which we are still developing, I just wanted to tell you why we are performing this study. Diffuse large B-cell lymphoma is one of the non-Hodgkin lymphomas, which is quite prevalent, and it is the disease of B cells, which are diffuse and quite large than normal, as shown in this histology slide. Most of the patients are treated with rituximab and a combination of chemotherapeutic drugs, CHOP, and 70% of them are cured. However, 30% of them would relapse from CHOP and go on and die. So the idea of this study is to develop a panel which will first sequence the diagnostic lymphoma biopsy, find out which are the driver genes which are aberrant in these lymphomas, and then follow in, once the patient starts treatment with R-CHOP, we follow these patients longitudinally with a response in their ctDNAs with the same panel. So the idea is to find out why these patients who are relapsing are, and what can be done, whether the driver mutations are still present, or what can be done to salvage these patients. So these patients, for this trial, we have already started standardizing, and we have tested around 82 samples. Some of these are ctDNA, but this work is still ongoing. But the third panel, the 56 gene myeloid panel, which we have been very successfully implementing with Professor George Vassiliou of Haematology, is to test the theory of clonal hematopoiesis in severe COVID-19 patients. And the samples we are receiving are from the Clinical Consortium of COVID-19 in the UK. We have already sequenced around 729 patients with severe COVID-19 disease to answer the question whether clonal hematopoiesis plays a role in this disease. And Ezequiel will now go through both the myeloid panel and the 350 cancer panel QC parameters with you in details.
Hello, my name is Ezequiel Martin. I'm the Senior Bioinformatician in the Cancer Molecular Diagnostics Laboratory in Cambridge, and I'm going to give you a brief overview of the QC of the data from the different clinical trials that Shubha has mentioned before. Just briefly, the pipeline, the bioinformatic pipeline with which all the data has been processed is just best practices from the different packages and software used, mainly the Broad Institute's GATK pipeline. And as a sample of the samples that we are going to be looking at, we are going to be looking mainly at germline and FFPE samples, being the main type of data that we analyze, but also some very representative ctDNA and fresh frozen tumor samples for a total of over 700 samples. As key exclusion metrics from the NGS data, the ones that don't present any problem, as you can see in our data, are the percentage of chimeras that tend to be sometimes a little bit high on FFPE data, but not in our TWIST panel, or the exclusion of base pairs due to mapping quality or base quality. The percent of PCR duplicates is remarkably different between the types of sample, being the highest in ctDNA, which is expected because it is very low starting material, and the lowest also as expected in germline and fresh frozen samples. Moving on to the percentage of overlap, which speaks about the fragment length of the fragments being sequenced. As you can see, it's very consistent, meaning that the fragments overlap in the center as you would expect, because of the fragmentation that is on the NGS protocol, except for the ctDNA samples, which you don't fragment with this, because it is already fragmented, so there is less overlap in the center of the forward and reverse read. And as for the off-target coverage, it's very consistently around 20%, which is very good because it is comparable to whole exome data. When we look at the correlation between raw number of reads on average in each sample and the final target coverage passing all QC, on average between all the samples, you can see that we have around 30 million reads of raw reads as input, and we get an average coverage output of 600. When you break that down by type of sample, as expected, the ones that perform the better and perform very similarly are the germline samples and the fresh frozen samples, while the FFPE is also as expected. The target coverage is slightly lower for a slightly higher input number of raw reads, and the situation it's a little bit worse with ctDNA, and that's why we sequence the ctDNAs to a higher number of raw reads, so we obtain on average the same final targeted average coverage of around 600. Looking at other important metrics of the NGS data, we have the fold 80 penalty base score, which speaks about the uniformity of the coverage across the whole panel. And as you can see, it's very consistently across of types of material of samples around 3.5, which is again on par with the fold 80 that you can see on whole exome data, which is the golden standard. While when you look at the insert size, as I commented before, on average on all samples is just below 200 base pairs. The lowest is the FFPE because the material is usually more degraded, while it is higher on germline and fresh frozen. When we look at Qqc Metrics on germline samples in the 350 gene panel, the largest one that we have in our facility, you can see that very consistently the PCR duplicates and the off-target are around 20% with very little variation. And when you look at the comparison between the input total reads and the final passing QC coverage, it's also quite stable. There are some samples which are outliers for different reasons, but it's quite stable overall. In the end, across germlines, we have around 23% PCR duplicates, 22% off-target coverage, and a final coverage passing QC of 660x. When we look at FFPE, the off-target coverage remains stable, which speaks about the quality of the design of the panel, while the PCR duplicates, they vary quite more, and it's also significantly higher because of the irregular and poorer quality on average of the FFPE samples. This is also true for the amount of raw reads that we get from the sequencer and the final coverage, with only a few samples in this pool of samples that come from several different clinical trials failing the minimum coverage filter that we have of 200x. This is the same kind of data from the smallest panel that we have in our facility, as opposed to the previous one, which is the largest one. And this is to show that, one, the panel works very well, also, but that as expected, the smaller the panel is, the larger the PCR duplicates become. In the end, you end up with more final coverage passing QC of around 1800x in the case of this panel, and that is on spite of the PCR duplicates being significantly higher because of the smaller size of the panel. In this myeloid panel, that is one order of magnitude smaller than the 350 gene panel, we end up with PCR duplicates of around 54%, of target coverage of around 27%, and a final coverage of 1732x. This is an example QC of one of the batch of samples that we do in CMDL. And in here, I want you to focus in this part of the graph in which you can see the curves of fragmentation of the different DNAs, and you can see how different they look, the pattern of fragmentation of the bad FFPE samples, the good FFPE samples being this yellow one, and the germlines, which are a smooth curve with the peak centered around 200 base pairs. If we look at the fragmentation patterns of more types of samples, you can see that ctDNA samples present their typical spike of fragments of around 150 base pairs, while the germlines, they have the smooth curve. And the ctDNAs, they also present this bumping here, which is the proportion, the portion of the plasma DNA that comes from the germline as opposed to the ones that come from the tumor. In here you can see another gradient of bad FFPs to good FFPs compared to the germlines, and in the last one you can see that as expected the fresh frozen tumor material behaves in an indistinguishable way from the germline samples. Going to have now a brief look at what at the variants that we call from the panel. On average, from the germline samples, we call around 3,000 genetic variations from each individual, which is quite what you would expect from a panel of 1.5 megabases in size. So no surprises in there. From the tumors, from the FFPE tumors, and this is also true for the fresh frozen tumors and the ctDNA material, you see that we call on average less than 500 mutations once you have subtracted the germline mutations which you do see on the tumor material. But there is a tail with a large number of mutations that represent the hypermutated tumors. Meaning the tumors that have more than 20 mutations per megabase on the panel covered territory. Speaking of the tumor mutational burden, we also call it accurately with our 350 gene TWIST panel. We have run a large validation study using data from other sources, but mainly from whole genome sequencing data, which is the golden standard to call tumor mutational burden. And we can call the tumor mutational burden in 72% of the samples, and in those 72% of the sample, we call the tumor mutational burden the status of high or low tumor mutational burden with a 94% accuracy, which is very good, because tumor mutational burden is becoming increasingly important criteria for inclusion in clinical trials. Finally, we also are able, because the panel is large enough, and the 350 gene panel is large enough, 1.5 megabases, to call copy number variation from the panel data using the G8K best practices pipeline. And we have validated this in a batch of samples using FISH as the golden standard for amplifications in this list of genes. And the genes that were amplified and the level that they were amplified to using FISH, we call with the 350 gene panel with 100% accuracy, all of them, while other NGS call amplifications calling in clinical setting, we call 63 of the 67 in this batch of samples, with no false negatives and with some false positive, which gives at ending a 97% accuracy on the calls of the CMVs compared to validated and FISH data. And that would be it. Thank you very much for listening, and any questions are welcome.